3 prime to 5 prime is template strand, okay, and then 5 prime to 3 prime is coding strand. Yes, this is template strand and this is the coding strand. And in between you have mRNA. The information from DNA is being transcribed to mRNA. You have understood, right? Okay. And one more thing I told you about the promoter and the terminator. Do you remember? Yes, okay. So when you just refer to this promoter, if they ask where is this promoter present, okay, then you have to refer it with the coding strand. When you see promoter, okay, so this promoter where it is present towards the 5 prime end of the coding strand. So wherever this 5 prime end you refer, you call this to be upstream. What do you call it as? Upstream. Okay. So where is this promoter present? It is present in the 5 prime end of the coding strand or 5 prime upstream of the coding strand. Always this 5 prime will refer to upstream. Then when you talk about this template strand, 3 prime, wherever this 3 prime present, you call that as what? Downstream. So 3 prime end of the template strand. Okay, here you use the term downstream. This is about what? Promoter. Can you understand? Guys, are you able to understand? There is this promoter present towards the 5 prime end or the 5 prime upstream of the coding strand. You have to refer with this coding strand. Okay, and then 3 prime end. So always this 3 prime here you call it as downstream. So when you talk about terminator, what is this terminator? Where it is present? 3 prime end of the coding strand. Again, this will, whenever you talk about 3 prime, it is what? Downstream. Yes? Okay, and then 5 prime end of the template strand. Oh, okay, right. So here, see 5 prime end, 5 prime upstream of the coding strand and 3 prime downstream of the coding strand. So that is why I have given you both. It is not mentioned. It's mentioned only about the coding strand in your textbook. Be clear. Always 5 prime strands refers to upstream. 3 prime refers to downstream. Here we are talking about promoter. When you talk about promoter, 5 prime end of the coding strand, which is the upstream, will be the promoter. When you talk about the promoter, you can refer even 3 prime template strand. Have you understood? One strand is being used as a template. Once she told me that if both the strands have the polarity 5 prime to 3 prime, right? So this cannot be used. That is one answer. Second answer, what did I tell you? In case if it is being formed, both will be coding for a particular protein for the trait. So that will not be possible, right? There is one more answer. In case if 2 strands are formed, both are single-stranded DNA. Then with the due to complementary base pairs, they might form a double-stranded RNA. Sorry, single-stranded RNA. So due to complementarity, what happens? Adenine pairs with uracil, uracil pairs with adenine, guanine pairs with cytosine. So they might develop double-stranded RNA. But this will not be possible because only one single-stranded mRNA will be involved in protein synthesis. Not all bacteria come under a prokaryote, right? Yes, they don't have well-defined nucleus. They don't have well-defined nucleus. Can you understand? So generally use the term cytosol. Normally we call it as cytoplasm. Is it not? Okay. So what happens here? Both transcription and translation takes place in the cytoplasm itself. Can you understand? Both transcription and translation takes place in the cytoplasm itself. But when you talk about the eukaryotic cell, what is a eukaryote? Plant cell, animal cell, all those which have well-defined nucleus. So here you have well-defined nucleus. They have membrane. Can you understand? Okay. So here you have nucleus and then followed by the cytoplasm. Cytoplasm, you would have learned about the cell organelles, mitochondria, lysosomes, and Golgi apparatus, vacuoles, plastids, endoplasmic reticulum. All these are cell organelles. Likewise, you have ribosome also. Okay, so here this is the ribosome. Okay, so this is the ribosome and this is the nucleus. You have segregation here. They have well-defined nucleus present in the eukaryote. So, transcription take place in the nucleus and translation take place in the ribosome. Again, go to central dogma, DNA, mRNA, proteins in the eukaryote, but the process is same. Transcription we have learned, okay, the same initiation, elongation and termination. The same process will take place in prokaryote with the help of only one enzyme. What is the name of the enzyme I told you? RNA polymerase. But one more difference I taught you. What about prokaryote and eukaryote? Here only one enzyme is involved. RNA polymerase, only one type is involved. Whereas here, you are learning, but what is that? 3 types. RNA polymerase 1, right? RNA polymerase 2, and RNA polymerase 3. So this helps in transcription of what? rRNA, ribosomal RNA. Here 2 helps in transcribing mRNA, and 3 helps in transcribing tRNA. Now, what is the main thing you are going to learn here? You remember this concept. Now we are going to learn in eukaryote. Prokaryote, I told you the same thing, initiation, elongation and termination occur, but inside the cytoplasm, then translation also takes place inside the cytoplasm. We'll come to that. Now we are going to learn about eukaryote. The 3 types of enzymes, RNA polymerase, 3 types are involved in this process. So what happens? What happens in eukaryote? Transcription in eukaryotes. So when we study about eukaryotes, the same first step, initiation, okay, the second step, elongation, and then the third step, termination. Okay, right. So what happens here? These 3 steps take place. What did I tell? Initiation, the same. Remember the transcription unit process. Always it will be double-stranded like this. Okay. And then the promoter is present here. The RNA polymerase binds with the promoter with the help of sigma factor. It joins them. It starts elongating. So when it elongates, this gets split up like this. Okay, and then the template strand, the information present in the template, it will be copied on to the mRNA like this. Strand will be formed, and then it gets stopped in the terminator region with the help of the rho factor. Can you understand? So this is what takes place here. Okay, now after this termination, it gets stopped. Now this RNA, the red color RNA, comes out from this process. Now, still it is inside the nucleus only. This is nucleus. This is nucleus. This nucleus what I have drawn here. Okay, still it is inside the nucleus. Once when it comes out of the nucleus, this is the cell, ribosome is present here. Okay, once when this nucleus comes, when this mRNA, this is the mRNA. When this mRNA comes out, it has to be a matured one. Use the term immature or not a processed one. Immature RNA, mRNA, or you call it as not a processed one. With this mRNA, what is present inside the nucleus, it is not ready for translation process. If it has to be ready, this should be a mature one. So once when it is matured, it will come out of the nucleus. Can you understand the concept? So here, there is another term I told you. Do you remember? HNRNA. For this, I gave you another term. HNRNA, heterogeneous nuclear RNA. Do you remember when I taught this? What is this HNRNA? It is the immature RNA. Okay, so when it becomes a processed one, you call this to be matured mRNA, which is ready for the protein synthesis. Then how do you tell this? This is hRNA. Okay, now I'm drawing it like this. Here in this mRNA, you have 2 regions. One is exon, the other one is intron. you have like this exons. What do you call this as? Exon. E-X-O-N. What do you call it as? E-X-O-N. Exons. Okay. And here, the red color I'm drawing it here. This will be introns. What do you call this as? Introns. I-N-T-R-O-N. Intron. And this is exon. Okay, there are two regions present here. They will be present alternatively. Exon, intron, exon, intron, exon, intron, exon, intron. It goes on. So this is the 5' end and this is the 3' end. So what are these introns? They are called as intervening sequences. Intervening sequences. As intervening. Okay, so what happens here? These introns are present between two exons. Introns are present between what? Two exons. So they intervene. Intervene is they are just present in between this. If these introns are present, then you call this to be a immature one or not a processed one, or this HNRNA you call it as, which is not ready for the protein synthesis. Have you understood? Introns from 5' to 3', but if this intron is there, it will not be ready for the protein synthesis. So what has to be done? These introns have to be removed. Introns have to be removed. So the first step, what is involved here is, you call it as splicing. What do you call this method as? Splicing. So what is this splicing? Removal of introns from the HNRNA, you call it as splicing. Now once when it is removed, so fully you will have what? After splicing, fully you will have only this exons alone will be present. Have you understood? So this is one step. This is the first step. Okay. Then next step is what? Second step, you call it as capping. What is the second step called as? Capping. C-A-P-P-I-N-G. Capping. What is this capping? This is the region 5'. Towards the 5' end of this HNRNA, you are going to add methyl guanosine residues. What are you going to add? Methyl guanosine. Okay, so here you will be adding here. I'll just mark it with some other color. This region. Okay, methyl guanosine phosphate. Methyl, M stands for methyl. G is guanosine triphosphate. That is why 3P is given. I told you removal of introns. Okay, now introns are being removed. Second step, you call it as capping. There towards the 5' end, methyl. M stands for methyl. G stands for guanosine triphosphate. Okay, these are added. So when these are added to the 5' end, you call it as capping. Something which gives protection for the mRNA. If this capping is not done, mRNA feels that it is unprotected. So anything might come and interrupt the process. Translation will not be able to occur. So 3 steps are needed to convert this HNRNA to processed mRNA or a mature mRNA. First step, we have studied about splicing. Their introns are removed. Only exons are alone present. That is first step. Second step, what is that? Towards the 5' end, methyl guanosine triphosphate is being added. Okay. Then last one is tailing. Towards the 3' end, polyadenylate residues are added. Now, next stage, last diagram you can see. This one you call it as messenger RNA or mRNA or matured RNA or processed RNA, which is ready for protein synthesis. I told you, after splicing, capping and then tailing, okay, this HNRNA, which is an immature one, which is not ready for the protein synthesis, it is being converted after the 3 steps to mRNA. Have you understood now?
If you like this video, share and subscribe my YouTube channel and follow me on Facebook and Instagram. Thank you.